Hi there, my name is Benton Willis and I'm a PhD student at the University of Florida. I'm studying a group of fungal pathogens called Exobasidium, which infect plants, including and closely related to blueberries and azaleas. These fungi can cause all kinds of symptoms on their host plants, including leaf spots or galls, which are tumor-like growths. These fungi do not kill their host plants, but they can harm their hosts in all kinds of different ways, which is just one of the reasons I'm interested in this group of fungi. I'm at Paynes Prairie State Park looking for some Exobasidium now. As I was walking along this trail, I noticed some leaf spots on this plant. Let's take a closer look. Unlike leaf spots caused by insects or other pathogens, I believe this abnormal growth is caused by an Exobasidium fungus because of the plant that it is growing on, which is a species of blueberry, and the light covering of white spores underneath. Unlike a lot of fungi that get their energy from dead wood or living tree roots and produce their spores in a mushroom, these fungi get their energy from the live plant and produce their spores on the outside of the plant tissue, hence the name Exobasidium, kind of like an insect has an exoskeleton. Species of Exobasidium can be found all over the world, on every continent. There is at least one species of Exobasidium described from Florida, and I think the one we just looked at might be a new species, unknown to science. I suspect there might be many new other species all across the US, so I need your help. I have a permit to collect Exobasidium fungi in Florida state parks, county and city parks, and select national forests across the US. If you have a permit to collect fungi or think you have Exobasidium fungi on your property, please contact me at caroline.willis@ufl.edu. I'm going to show you how to collect these fungi, make a spore print, and send me a sample so that we can have a better understanding of these fungi across the US. In order to prevent further harm to the plant, we are going to use proper pruning technique in order to collect the fungi and ensure proper healing of the plant. Looking at the gall or branch in question, follow the limb to the point where it connects to the rest of the plant. Clip here, and the branch will seal itself off, and it will look like we are never even here. Then, place the sample in a clean plastic baggie. This will keep the material from drying out and fresher while in transit. If you would like to stop here and have access to a FedEx location, please send me an email and I can send you a FedEx label at no cost to you. If you would just like to send a spore print in an envelope via USPS, keep watching. When I collect Exobasidium in the field or receive samples by FedEx, I can get a spore print directly onto growth media in a petri dish. But I can also analyze the spores and get the fungus into culture using a simple spore print technique on tinfoil. I'm back at the lab, but I'm going to show you how to do a spore print technique at home. Depending on your material, either place an entire leaf or a portion of your gall in the center of your tinfoil face down, so that the spores or the white powdery area is facing the tinfoil. Place a clear glass or Tupperware over the tissue to prevent any airflow from blowing the spores away. Because these spores are white, circle it with a Sharpie so that I know where to look when I get your sample. Leave it at room temperature for 6 to 12 hours so the spores have time to release. I recommend setting a timer so you don't forget the next part. Once your sample has finished printing, take away the covering and carefully pull away the gall. You should be able to see a light covering of white. Let the tinfoil air dry for 10 to 20 minutes so the spores do not get moldy in the mail. Fold up the tinfoil, protecting your carefully made spore print. You can then place the sample in a simple envelope, attach a stamp, and use the address to send it to me below. If it ends up being something interesting, I may ask you for additional samples or information. If you would like to air dry the rest of your sample along with a portion of the plant, I encourage you to do so. And if you have any other questions or additional concerns, please let me know using my email. Thank you and happy collecting! If you would like more information about how to identify Exobasidium fungi and how to help me with my research, please scan the QR code or copy the link in this video to see my written collecting protocol. And if you haven't already, I also heavily recommend making an iNaturalist account, taking lots of photos of the fungi you observe, and writing your iNaturalist record numbers on the sample bags or the spore print tinfoil, so I have complete data for the samples you send to me. Observations made on this platform help scientists like me find the organisms we study and give us a better idea of the biodiversity in the world we live in. If you do not have reliable access to internet or prefer to send me spore prints in a plain envelope, please include your name, collection date, location, which we will obscure in a public database, and if possible, the name of the host plant. Thank you again, and if you still have questions, please do not hesitate to reach out at caroline.willis@ufl.edu.